Yeah, so it's my pleasure to speak with you all today. Just a matter of additional background. I'm an organic chemist by training. I see a lot of experimental chemistry in this talk. And what my group is really interested in is, from a chemistry perspective, is reaction discovery and catalyst discovery. But as we've gone sort of further and further into that, we've been very interested in the integration of machine learning tools with experimental chemistry to facilitate reaction discovery and catalyst discovery. And so what I thought I'd do today is share a little bit about the workflow that a chemist typically approaches when going forward in a reaction discovery or a catalyst discovery campaign, and then talking through some opportunities to integrate machine learning to make that process either more efficient or discover better solutions than one might otherwise be able to achieve using standard tools. Okay, so here's an example of how my group would go about developing a new reaction. Typically, we're going to have some mechanistic hypothesis about the type of chemistry that we want to achieve, the type of products that we want to develop. In this case, my group is interested in nickel photoredox catalysis, and so we might have some mechanistic idea how we could sort of achieve a new CC bond forming reaction. Now, we need to go into the lab to sort of realize this in practice, and to do so, we need to do a few things. First, we need to define a model system such that we can sort of experimentally assess the feasibility of this process. So in this case, that means choosing a substrate or substrate partners. It means choosing an initial guess of conditions that usually requires the literature as well as really relying on your expertise and prior knowledge. This turned out to be a pretty effective, initial guess, in that we got a 50% yield. So this was a great initial start. From there, we need to optimize the conditions, and typically chemists do this, sort of one factor at a time, looking at multiple dimensions like solvent and catalyst loading and catalyst identity. And so in this particular case, we did that. We looked at a whole slew of different variables. We probably ran about 200 or 300 experiments to sort of optimize sort of this particular system. And then we want to sort of assess how good those conditions are across a diversity of chemical space. And we do this because when we started off, trying to discover this reaction, we didn't necessarily want to make that specific product, but we wanted to establish proof of concept that this catalyst system works. And now we want to show that it will work for the types of substrate partners that practicing chemists would be interested in. So for example, if you were a medicinal chemist, that you could adopt this chemistry and it would work. So then we choose sort of a whole host of different substrates to test our optimal conditions with. And then hopefully you go ahead and publish such a paper. In this case, you can see sort of the reference down here. So if we think about this from the lens of what the goals are in this process, I think you'll see that, or at least I see, there are a lot of issues with the way that we go about this reaction discovery process. The first one I'll just point out is this: how we assess sort of our conditions. The goal, as I said, is to try and give an outside user an idea of how these conditions will fare on an unseen substrate. And so what we want to be able to do is assess a variety of different substrates such that ideally you can sort of give a chemist that intuition or predictability, right, for something that's unseen. But we don't do a very good job of this. This has been sort of talked about in the literature quite a bit, partially by the pressures of publishing. We often are going to report high performing substrates as opposed to ones that are really diverse. So there are ways in which we can still sort of assess, sort of how robust the conditions are. There's beautiful work from Frank Lorius's group saying that even though experimentally it can be very difficult to evaluate many, many different substrates, you can choose one substrate partner and look at additives and see how functional group tolerant the reaction is. And this can give you some sense of the robustness or tolerance of a synthetic method. Scientists at Merck have also sort of pioneered this idea of informer libraries. These are libraries of really complex molecules that have medicinally relevant functional groups in them. And what they hope is that people will assess these types of substrates, and this will give medicinal chemists or other practicing sort of chemists a better sense of how effective the reaction conditions are. A few years ago, we sort of thought about an alternative approach that could sort of address this issue, and this was building off of a lot of important sort of work that was developing in the data science and chemistry field from folks like Natalie Fay and Matt Sigman and others, wherein we thought we could use unsupervised learning techniques in order to help sort of chemists generate more representative and unbiased substrate scopes. The idea would be to take, all commercially available examples of a given substrate class. You would then do some sort of, filtering if they're certain, if you want them all to be, low cost and available from a certain supplier, for example. We then featureize these using density functional theory. We do this so that we can capture the steric and electronic properties of these substrates that would allow us to sort of predict similar reactivity and similarity between discrete components. This, of course, requires some sort of high throughput sort of DFT featurization approach, and my group has developed this process called AutoQChem that allows us to do this reasonably efficiently. And all of our sort of libraries of molecules that we've computed and their DFT features are available on an open source sort of web app that you're welcome sort of to take a look at down here. Then one can sort of cluster these substrates based off of their features, such that sort of one can sort of come up with chemical space maps where you can select maximally diverse or sort of representative substrates that cover that space. And this hopefully can lead to sort of less biased evaluation of the generality of a specific set of conditions in chemical space. However, this approach doesn't actually change, the conditions that you've identified in sort of the process of optimization. And so it's very possible that, we have a sort of often saying where you get what you screen for. If you only looked at a single model substrate when you're optimizing, the conditions may not fare well across, a highly sort of diverse space. So are there ways, if we're seeking sort of conditions that are maximally sort of general, in which we could sort of change the way that we do optimization to sort of get at this, this goal? So that's a question that the field has been sort of asking, especially recently, although sort of historically chemists have had approaches to this as well. And there's a really nice example from the Jacobson lab that was published a few years ago, where they looked at screening instead of a single model substrate against many conditions. They actually screened 14 different sort of products, substrates against catalysts in this case. And what they found in sort of doing sort of this high throughput experimentation is that indeed the catalysts that sort of showed up as most general, like this one sort of, it's a vertical row, this one right here, is not one that had been sort of recognized as a good or optimal catalyst based off of some of sort of the discrete substrates that had been sort of used for optimization sort of previously. So this really nicely highlights, the importance of looking beyond a single model substrate in optimization. But analytically, this is a real sort of challenge, both, being able to have, the material and time and resources to do sort of an evaluation across many substrates, but also, especially in this case where they're looking at EE, being able to sort of assay for sort of EE in this larger chemical space. So I think what many of us have been thinking about is, are there sort of data efficient approaches to doing this, especially when one doesn't want to just evaluate one reaction dimension like a catalyst or a ligand, but many dimensions like catalyst, ligand, base, additive, and so on. Soon, if you're trying to do that alongside also evaluating many different substrates, this becomes experimentally intractable. So there's a beautiful example from Marty Burke and Alana Spiraguzik and Bartosz Grabowski that was published a few years ago that attempted to do this using Bayesian optimization, wherein they were looking at a Suzuki cross-coupling, 11 products in 48 conditions, and they sort of adjusted sort of a Bayesian optimizer in order to be able to sort of do active learning in this space to find more general conditions for the Suzuki coupling. And so you can see sort of in the process of combining sort of uncertainty-based Bayesian optimization with robotic experimentation, they were able to find sort of more general sort of conditions. This was the benchmark, and they ultimately found better conditions across sort of multiple substrates by surveying about 50% of the condition space. Around the same time, my group was interested in sort of pursuing a different ML approach to doing data-efficient identification of general conditions. Our team is a sort of a big one and an awesome one. It was led by Jason Wang, who's a fifth-year graduate student in the group, and he was joined by a number of coworkers in my lab. And this was an incredible collaboration with scientists at BMS, led by Jay Stevens and Christine Golden and June Lee. And the idea that we had, which was really an idea from Jason, was to think about this in terms of multi-arm bandit optimization or reinforcement learning problem. In the classic formulation of a multi-arm bandit, is sort of the setting is an agent is going to play slot machines, which are known as one-arm bandits. And when you're playing slot machines, your goal is to maximize your reward and to identify the best slot machine. But of course, when you have a certain budget, right? But when you start off, you don't know what the probability distribution is of these slot machines. And so you're going to need to explore them. And then as you learn something, exploit the ones, that have the best payout distributions in order to maximize your reward. And what Jason saw as an analogy to sort of this generality optimization problem, where if you imagine that these slot machines are now conditions, in the simplest case, let's say that there are different ligands, and the distribution is the distribution of yields or anti-selectivity or both, across a substrate scope that you've defined. So like in sort of the classic formulation for playing slot machines, the idea is you want to identify the best condition, best being the one that has the highest distribution of high reactivity across the most number of substrates, and do so in the least number, of experiments, so within sort of a minimum budget. So Jason went ahead and started to evaluate this first using sort of some simulated data where he could compare against traditional A/B testing, the bandit algorithms, to sort of benchmark and see if this actually sort of could find the most general conditions and in what sort of level of efficiency. So ultimately, he implemented over 25 different bandit algorithms and benchmark these with our synthetic data using both binary rewards, which is the most common formulation for bandit algorithms, but also continuous rewards, which are much more common in a chemistry setting where you have yields, that are distributed across, from 0 to 100% or ERs or delta delta G double daggers. And most of these algorithms are, if you're familiar with Bayesian optimization, have sort of a similar type of sort of balance between sort of exploiting and information that one has and exploring areas of high uncertainty sort of from the algorithm. This is just one example of a relatively simple, but what we found to be a quite effective algorithm, wherein you're sort of doing sort of that sort of balance of exploitation and exploration. And so the workflow here that we implemented sort of is shown in the schematic. So the idea here is that the chemist is going to define the substrate scope, the collection of substrates that one wants to evaluate, as well as the conditions that one wants to optimize. Then what we're going to use is one bandit algorithm that's selected based off of some sort of experimental assessment that I'll show you in a few slides. It's going to select which conditions to try and for which substrate partners. So that's sort of the first sort of suggestion, the conditions to evaluate and the substrates to evaluate them on. The chemist will then run those experiments, put the labeled data back into the algorithm. The algorithm will select the next conditions to evaluate on the next substrate, and you'll sort of iterate from here. We also have sort of a prediction model that's going to be just a simple random forest model that sort of accompanies this workflow. And the goal of the prediction model is to allow you to do batch-wise suggestions of experiments if you don't want to run one at a time, which we tend not to just from a time perspective for running these experiments. Okay, so we sort of statistically assessed a lot of these sort of combinations in some existing data sets, but the truth is that there aren't very many good sort of existing experimental chemistry data sets to use for this type of assessment, because what you need is a data set that's sort of diverse in substrates and in conditions, and most HTE data sets that are available are usually a single product in diverse conditions. So we built a few of these, ran a few of these as tests for this algorithm. I'm just going to show you one example. So this was a HT data set that we generated. It was a palladium catalyzed imidazole arylation reaction. We were going to survey 24 different phosphine ligands and 64 distinct products. This was a huge experimental undertaking because we had to, for the sake of generating the HT data set, isolate and make all 64 products so that we could generate calibration curves in sort of the process of the HTE. But Jason and Jay did this. This is the outcome of the HT data set. Here the big squares are the phosphine ligands and the little squares are the 64 distinct products. And since we had this data set, we were curious, would a model system-based approach to optimization have worked, in finding sort of the most generally applicable phosphine? And the answer is no, What we found, interestingly, is that, so this is looking at each distinct product, and I'm color coding which phosphine was optimal for those sort of distinct products. A lot of them had low yields, but the ones that had higher yields, you can see many different phosphines showed up as optimal. So there's no single phosphine that's optimal, and you'd get a different answer depending on the model substrate that you identified, rather than identifying what the most generally applicable phosphine might be. When we deployed the bandit optimization algorithms in this space and sort of assessed, ran a few simulations to sort of compare different algorithms as well as against sort of baseline, pure exploration as well as sort of an explore then commit strategy, we saw that in particular, the Bayes UCB and UCB algorithms outperformed, baselines, and typically sort of allowed us to identify the most general conditions in about sort of 80% accuracy within 10% of the search space. And we saw this for a few different data sets, so that became sort of our goal and benchmark for deploying this in sort of real-world settings. Okay, so I'm going to just show you one real-world application of this in the context of amide coupling reactions. So I'm not going to sort of spend too much time on the background here, but amide couplings are one of the most important reactions in medicinal chemistry. It's shown sort of in this survey from 2014, so a long time ago now, but it's at that time was the most frequently used bond-forming reaction in med chem. And because of that, there's a lot of understanding and data on what conditions, one medicinal chemist use to do amide bond formation. And you can see sort of in this pie chart that the activator HATU and either triethylamine or diisopropylethylamine are the most frequently used conditions for amide coupling. So a question you might ask is, because they're the most frequently used, are they the most general conditions that for a particular library of amide coupling reactions? So that's exactly what we wanted to ask. We wanted to design sort of a fake library, if you will, where we're going to take indomethacin, this carboxylic acid, generate a library with a variety of different anilines. Anilines tend to be harder, right, substrates for amide coupling just because of reduced nucleophilicity. So we wanted to make it a bit of a harder, right, problem in that regard. And so these were the anilines, the 10 anilines that we chose. We chose these using that workflow I showed you in the early part of the talk, where we took all commercial sort of anilines. We did sort of featurization of them. In this case, you're just seeing a PCA plot. And then we chose 10 based off of 10 distinct clusters. So these sort of represent some sort of diversity in aniline space. The conditions that we chose to survey through the bandit optimization are shown here. So there are a variety of different mechanistically distinct activators that we chose, 3 different solvents and 4 different bases. And this was based off of some of that survey of what are the more commonly used activators, bases, and solvents. Okay, so it's 96 conditions. We then deployed the bandit optimization, so using the CBB-tuned algorithm to select the conditions. We're going to do 5 experiments per batch using the random forest algorithm as our prediction sort of task and sort of the Krieging believer sort of hypothesis to allow for batching. So this is the first set of activators and bases, substrates, and solvents that were suggested to run. We ran them. We fed the experimental data back into the algorithm. Right, it suggested the next conditions, to try on the next substrates. Now you can see the random forest sort of predictions and our experimental yields we fed back in. And based off of our sort of prior sort of work, we knew that we were aiming to sort of do this to survey 10% of the search space, and at that point to see which conditions were predicted to be most general, both based off of the random forest model, but also what the bandit algorithm is selecting the most frequently as the condition to try. So when we did this after sort of 8 rounds, so 40 experiments, we just sort of quickly sort of looked at what the model rankings were, and we saw sort of these 4 activators were ranked higher than sort of the other 4. This was interesting because HAT2, right, which I had said is sort of the most frequently used activator, is down here sort of in the bottom. We then continued to do the optimization for a total of 16 rounds. And the model rankings for the conditions sort of ultimately sort of are shown here. This was really interesting and exciting, especially to our BMS colleagues. They're more familiar with peptide coupling, amide couplings than we are, but the fact that this DPPCL activator showed up as sort of the top ranked condition was not expected. And so we wanted to then sort of go ahead and sort of assess this set of conditions to see, in fact, is it more general than 2 benchmarks, the HAT2 diisopropylethylamine case, as well as a set of conditions that our BMS colleagues had identified for challenging sort of amidation reactions sort of previously. And so here's the assessment, and what we see sort of generally is our top 2 conditions outperform or are as good as these benchmarks for all of the sort of tests that we sort of have run, which was really exciting. We also just wanted and were curious to know what the ground truth was in that data set. We only ran 120 experiments, and this was what the model rankings were, but how does that compare to the ground truth? So to do so, sorry, I'm going to skip over this. We actually did run the whole high throughput experimentation sort of data set for this amidation, and then could compare the true rankings to the model rankings. So here are the true rankings on the right for sort of the data. And what one can see is even after 40 experiments, the model had gotten the top activators correct, DPPCL and BOP chloride. And after the 120 experiments, the top 4 conditions were ranked correctly without having to survey that whole search space. Interestingly, again, sort of HAT2 is not the most generally applicable based off of average yields as a metric, condition, and that's also something that the model picked up on, even after surveying just 5% of the search space. So this was really exciting to us. We've gone on to sort of apply this in other contexts, but I just want to sort of conclude here given time. I think it's quite clear that a model system-based approach to reaction optimization is really unlikely to identify the most generally applicable conditions. If that's your goal, then this is not a good approach, right, to doing so. And so sort of, being able to first think about sort of the chemical space of substrates that one wants to sort of identify a catalyst or a reaction for, and then optimizing, right, is going to be a much better solution, right, to that problem. However, it is experimentally sort of costly. And so we are seeing, right, that these reinforcement learning models like a bandit optimization can be a more data efficient approach to this. Now, it's still not very data efficient, right? 10% of a search space, if that search space is millions of conditions and substrates, is just still not experimentally feasible. So we're certainly trying to think about ways to make this more efficient. Right now, we treat the arms as independent, those conditions, but there's ways of doing contextual bandits that might lead to better sort of data efficiency, and that's something that we're looking at right now. Okay, so with that, I just want to end by thanking sort of my group. I've only really talked about sort of one project, and that's sort of a project that Jason has led, but I have an amazing group of graduate students and postdocs and undergraduates, and I'm really grateful to have the opportunity to work with them and get to know them as people. It's really sort of incredible sort of privilege to get to spend my days with them, learning from them and with them. And then want to call out BMS colleagues. They've been an amazing team of collaborators, really indebted to their intellectual and experimental contributions to our projects, as well as the NSF Center for Computer Assisted Synthesis, which has funded this work. And I'd be happy to answer any questions.